Good evening, my dear student. Myself, Dr. Sneha. We are going to discuss about the topic today, tracer techniques. So in the tracer technique, we will see the introduction, then various techniques, then how we have to introduce all these radio label compound into the plant. All these things we will start and we'll study in detail. See, what is this tracer techniques first? Most of the time, what will happen? All these plant, they'll prepare their own food material. It will be utilized for their own growth, but at the same time some byproduct or the secondary metabolites or waste product will form when they will prepare their own food material. So these waste products are nothing but a secondary metabolites, and most of the time they will be useless to the plant, but they will have the pharmacological activity. So most of the human beings are all we are using these secondary metabolites as a drugs. And each and every plant or medicinal plant is having capacity to synthesize all these secondary metabolites. Now, how these secondary metabolites will form in the medicinal plant, that we will study with the help of tracer technique. So there are so many techniques which we will use to study the formation of secondary metabolites, means which metabolic pathway is used by these secondary metabolites to produce, which are the actual metabolic pathways through which they are getting, means which amino acid is used, okay, then which pathway it is going, and finally we are getting alkaloids, glycosides, tannins or volatile oil. All these things will be studied in the tracer technique. If you will see, all these things are coming under the biosynthesis. See, there are two things. Synthesis will be there, biosynthesis. Synthesis means what? Chemical synthesis. Any compound will be synthesized into the laboratory is a chemical synthesis. But as we are using here medicinal plant for synthesis of the secondary metabolites, it is called as the biosynthetic process. Means bio in the sense biological system. In the biological system, that is nothing but a medicinal plant, so many secondary metabolites will be synthesized and they will have certain pharmacological activity. So that is the biosynthesis. It is nothing but a process of making the complex product from simpler component through the chemical reaction at the cellular level. Means what? Primary metabolites are as it is natural compounds or the metabolites which we are getting from the soil will be utilized by the plant. They will use the sunlight, they will use the carbon dioxide, they will use the water, they'll prepare their own food material. In the plant itself, they will synthesize all these secondary metabolites, is called as the biosynthesis, means synthesis of the secondary metabolites into the medicinal plant. So this is the general introduction. So whenever we have to see the biosynthetic pathway, means how these secondary metabolites will be synthesized into the plant, these three techniques mostly we will use. Which are the methods? First will be the grafting method we can use to analyze how these secondary metabolites will be synthesized. Then mutant strain technique we can use, and tracer techniques. But as per the syllabus, tracer techniques we have to study in the detail. So grafting method and mutant strain method is also there, but so many data is not available, and nowadays we are not using this method. But just introduction about the grafting method is, see, what is grafting? Everybody knows. One plant will be grafted on the another plant, and then new plant, whatever we will get after grafting, we will see which active compounds or the secondary metabolites will be there in that new grafted plant, that we will try to see. One example I will tell here. See, tomato shoots will be grafted on the datura. What is there? See, datura is mostly having capacity to synthesize the tropin alkaloids. So mostly these tropin alkaloids will be synthesized into the datura plant. But if we'll graft this tomato shoot on the datura, we have to see whether this tomato shoot will get any tropin alkaloids or not. So this is the thing we have to study in the grafting method. See, if tomato will get higher amount of tropin alkaloids, what it indicates? Shoots of the plant is having capacity to synthesize the tropin alkaloid. But in this case, that tomato shoot did not get higher amount of alkaloids. Means what indicates, as we have grafted tomato in the datura, and second time again we will reverse the thing. On the tomato plant, we'll try to add the datura plant. So that time, which plant is having more amount of this tropin alkaloid, that we'll try to see. See, datura plant will have more amount of alkaloid, means it indicates that roots of this datura plant is only having capacity to synthesize the tropin alkaloid. So this is the grafting method. Here what we will do? So one medicinal plant parts will be grafted on the second another plant where they'll be nearly correlated. Then we will try to separate the alkaloids or many major active compound from both the plant, and we will try to see the quantification. So if it is more, then we can directly conclude that the, like example here is datura is having more capacity to synthesize the tropane alkaloid. Now next is the mutant strains. See, what is the meaning of mutant strains? There are certain microorganisms or some strains of the medicinal plant will be formed as it is because of the nature or something else. So these mutant strains may have capacity to synthesize certain types of secondary metabolites. So these type of studies also will do. If naturally mutant strains will not form, what we will do? We will take certain types of microorganisms that will try to add into the medicinal plant. So these microorganisms will get capacity to do the changes in that particular medicinal plant mutation, and then certain types of secondary metabolites will be synthesized. So example is here lactobacillus. When we will add into the mevalonic acid pathway, it will increase the biosynthesis of isoprenoid compound. Now isoprenoids are nothing but a volatile oil compound. Means what? Whenever we will add this microorganism in the medicinal plant, it will enter into the mevalonic acid pathway and it will try to increase the biosynthesis of volatile oil compound, that is isoprenoid compound. So this is the second technique, and most important technique which we have to study in the detail is tracer technique. See, what is tracer technique? It is nothing but just we have to trace out the biosynthetic pathway for each and every type of secondary metabolites. Now what is this tracer technique? In the tracer technique, most of the time we'll use radio label compound. So all these radio label compounds will synthesize first, whether it is obtained from the nature or we will prepare that. Then that compounds will be added into the medicinal plant. Then they will enter into the metabolic pathway. Through that, again they will be involved in the final synthesis of the secondary metabolites. So that secondary metabolites will be isolated and radioactivity will be compared, or it will be identified. So this is the general introduction about the tracer technique. So most of the time, if we'll see the definition of the tracer technique, it is a technique where we will utilize the radio label compound to find out or to trace the intermediate compounds or exact biosynthetic pathway for synthesis of any type of secondary metabolite, but for the particular time or at the particular rate. So this is only the definition of tracer technique. It is nothing but a technique where we will utilize the radio label compound to trace or to find out the different types of intermediate compound or biosynthetic pathway. So in this technique, so many secondary metabolites will be studied and we will be incorporating the radio label compound. So there are two types of radio label compounds. So radioactive isotopes will be there, stable isotopes will be there. Now what is the meaning of radioactive isotopes? So these are the radioactive isotopes are nothing but the isotope compounds we will prepared by the emission of certain types of radiation, either alpha radiation, beta radiation, gamma radiation. We will bombard on certain elements like carbon, hydrogen, sodium, potassium, sulfur, and then whatever radioisotopes, stable isotopes we are getting, that will be using. So that, but their half-life should be more. That is nothing but a radioisotopes. So most of the time, in order to study the biological system or medicinal plant, we will be using carbon and hydrogen. If we have to study the metabolic pathways of any medicinal plant, we will be using sulfur and phosphorus. Or if we have to study the proteins, amino acids, or any alkaloid, then we will use mostly the nitrogen compound. So these are, but all these radioisotopes, whatever we are getting, condition is they should be stable. If their half-life will be more, then only we will use these stable radioisotopes to study the metabolic pathway. Now, stable radioisotopes. So what is the meaning of this? As it is, we will not bombard or any element with the this alpha particle or gamma, that is radiation. What we will do here, as it is in the nature, whatever stable radioisotopes we will get, that will be introduced into the medicinal plant to study the biosynthetic pathway. So example is hydrogen we can use, carbon we can use, nitrogen, oxygen, anyone, but it is both will be same. But here in the radioisotopes, we will be bombarding with hydrogen atom with the radiation. Whatever compound we are getting, that will be using. But here, naturally occurring stable isotopes only we will use. So these are certain examples. Now, why we have to use this? What is the significance of this tracer technique? Actually, this is very like highly sensitive method. So in this method, what we will do, as we are preparing the stable radioisotopes, we'll be introduced into the medicinal plant. So at any stage we can use. Any stage, in the sense, in between the biosynthetic pathway, if we study, we will get idea. So there we can introduce, or during the when we will add all these primary metabolites to the medicinal plant, that time we can add. So that and whatever compound we are getting, that compound will have the higher amount of this radioactivity. So it is very sensitive method and highly sensitive method, because it is totally depend on the radioactivity of the compound which we are introducing into the medicinal plant. After that, again we will try to separate or isolate the secondary metabolite, and we will check the activity. So that's why it is very sensitive method, and it is applicable mostly to all the living system. How it is applicable? Because each and every type of radioisotopes we can be introducing into the medicinal plant by various ways. So any plant we can use this technique. Then a wide range of the isotopes are available. You can see, for if it is a nitrogen containing compound, like alkaloids will be there, proteins, amino acid, nitrogen we can use, see, like that. And hydrogen is present in all the structures, so hydrogen also can be used. So there are wide range of the radioisotope compounds in the nature, that is stable one, or if stable one we are not getting, also we can use the radioisotopes. And this is more reliable method and easy to administration, means all these radioisotopes can be easily administered to the medicinal plant, and rate of isolation also it will be simple. Then just we have to check the radioactivity, and we can trace the tracer technique, means whether same biosynthetic pathway will be followed by the same compound with the same protein or not, that easily we can see. And most of the time, or 90% time, we will get the accurate result. But condition is isotope should be stable. It should not undergo the degradation. It should not harm the medicinal plant. If this condition will be followed, we'll get the 90% correct result with the tracer technique. Now, what is the criteria here? So starting concentration of the tracer. Tracer is what? Tracer is nothing but a radioisotope compound. So whatever radioisotope, like carbon will be there, hydrogen, nitrogen, whatever we are using, no, it should be stable. It's with a low concentration also, it should enter into the medicinal plant. So that is the first criteria in the tracer technique. It should not be like we are having the higher amount of that like concentration of the stable radioisotope, and it is not entering into the plant. Then that time we will not use this. At the lower concentration also, this tracer compound should enter into the medicinal plant. That is the first condition we have to follow. And physical and chemical nature of the compound also matter. See, if it is a radioactive compound, means it should not harm the medicinal plant. If we will enter and that plant cell will die, then it is useless. Means what? Physical and chemical stability of the radioisotope also matters. It should not harm the cell where we are introducing this radiolabel compound. That is the second condition. Now, third condition, we are having stable compound. It is not affecting its physical and chemical nature, but thing is, it should involve in the biosynthetic pathway. It should enter into the pathway. If you will have all the condition, and if that radio compound will not enter into the synthesis, then again it is useless. So third condition is, whatever radiolabel compound we will introduce, it should directly enter into the biosynthetic pathway. That is the third condition. And last one is, when it will enter, it should not damage any intermediate compound when it will enter into the biosynthetic pathway. If it will damage, what will happen? Same biosynthetic pathway won't be carried, and at last, secondary metabolite, whatever we plant, it will not be synthesized by the medicinal plant. So these are the four conditions or the criteria. For that also we have to think for the study of this tracer technique. Now, advantages are, this method is highly sensitive, is applicable to all the living organism, wide range of the isotopes are available. This is more reliable method, because mostly carbon, hydrogen, oxygen, nitrogen, these compounds only we are using as radioisotopes. Easily they are available, and each and every structure of the secondary metabolite, these compounds are available. So it is very simple and easy method, and most of the time it will give the accurate and current result. But now there are certain limitations also. Now, what is the limitation? Each and every radio compound will have some kinetic effect, some chemical effect will be there, some radiation effect will be there. Then radiochemical purity of that compound also matter. See, there are certain kinetic effect, chemical effect, and radiation effect. They may affect the biological activity of our secondary metabolites, or they may damage the cell. Sometimes they may change the intermediate compound. All these are the limitations. So if we'll think for that, last and most important limitation is radiochemical purity also. Sometimes what will happen, one compound, if we'll take an hydrogen example also, hydrogen may have 2 or 3 types of isotopes. In that case, whatever isotope we want, like H2 or H3, if you want H2, if H2 compound only should be there with a higher concentration, means radio label compound, stable one, it should be only H2. But whenever we will bombard hydrogen with any radiation, H3 may form. So if concentration of H3 radioisotope will be more, it is useless for us. That's it, the radiochemical impurities matter here. If any impure compound, impure in the sense impurity in relation to the other radio label compound will be there, it will affect the, again, tracer technique, or it will affect the activity, or we will not get the exact study whatever we have planned. So all these are the limitations to the tracer technique in relation to the radioisotopes. Now, there are certain steps how to do the tracer technique. First important point is preparation of labeled compound. How to prepare the labeled compound? Just I told there are two methods. First is stable radio compound, and second one is with the radioisotope. Means radioisotopes compounds will be prepared with the help of radiation. We will bombard that compound with alpha, beta, gamma radiation. We will get the radio label compound. Stable radio compound can be used into the study. Or second method, in nature, whatever stable radio compound we are getting, same radio label compound can be used and it will be introduced into the medicinal plant for further study. This is the first step in the tracer technique, preparation of radio label compound. Here, either naturally occurring stable isotopes we can use, or with the help of radioisotope, or with the help of radiation. But condition is same, stable radio compound we should get, or isotope we should get. Now second is introduction of the label compound into the biological system. Now we are having the stable radioisotopes. We have to introduce that into the biological system. Now these are the techniques. With the help of this, we have to introduce all these radio compounds into the biological system or the medicinal plant. Now what is this technique? First will be the root feeding. In this, what we will do, whatever plant we have selected for the tracer technique study, we will prepare the solution of this radio compounds or radioisotopes or radio label compound. Through this root, this in the liquid form will feed into the medicinal plant. So what will happen? As we know, roots are having capacity to absorb the water from the soil. So through that water, these radioisotope compounds will be going into the plant body. Then from there, it will go to the site of action. Site of action in the sense, if leaves are having capacity to synthesize the secondary metabolites, all these radioisotopes will be carried to the leaves, where they will involve in the biosynthetic pathway, and then whatever secondary metabolite will be formed, it may deposit either in the leaves or any part of the plant. That is the first method, root feeding. Here, radio label compound will be introduced into the medicinal plant through the roots only. First, we have to prepare the solution. Through the root, it will go to the site of action. This is the first one. Second one is the direct injection. In the direct injection, we have to see at which place or in which organ of the medicinal plant that secondary metabolite is getting synthesized. At that place only, we have to introduce our radio label compound solution, same place. Like if it is a stem, suppose synthesis of the, like we will take an example of sandalwood oil. Sandal oil or volatile oil of the sandalwood will be synthesized into the stem. So whenever we have to introduce any radio label compound into the volatile oil, we'll give the injection to the stems. If it is for the this root, we will give through the injection directly to the roots. Or if it is a leaf, we'll give to the leaf only. Now next method is the floating method. So here what we will do, most of the time, leaves will have capacity to absorb the moisture or water. In that case, we will prepare the solution of the radioisotopes. Then whatever part of the plant is having capacity to synthesize the secondary metabolite, that plant will directly dip into the solution of radio label compound. So whenever they'll come in contact with each other, so that radiolabel compound will be entered into the plant body and they will be distributed whole plant body, and wherever it is required they will directly enter into the biosynthetic pathway. That is the floating method. Now stem feeding is there again here. In the stem feeding, if any secondary metabolite will have capacity to synthesize the secondary metabolite in the stem, so directly through the injection or through the pipe or through the line also, just like that we can feed our radiolabel compound into the stem. Again, through the stem, it will be carried to the various parts of the plant. As we know, all the stems or the roots or the bark, they will have this xylem and phloem cells. So through that, it will be carried to the whole plant body. Now infiltration. So infiltration is again another technique. In this, what we will do, as it is, we will not dip in the solution. If suppose we have to feed radiolabel compound into the stem, what we will do, we'll take the thread like thing. We will have the beaker or something container. In that, we will take the thread like structure. Thread will be dipped into the beaker and same thread will be enter into the stem. So that radiolabel compound in the solution form, it will be carried through that thread into the stem of that particular plant, where radiolabel compound will be introduced into the stems or any part of the plant. I have given the example of stem. If it is a root, same thing we have to repeat for the root introduction. So that is infiltration, means as it is, we will not keep our radiolabel solution in contact with the part of the plant. We will use something else. That is nothing but either thread we can use. So through the thread, that liquid will be transferred into the plant body, and then through that it will enter into the metabolic pathway. Now, next technique is the spray technique. Spray technique mostly will be used only for the aerial part of the plant. Aerial parts of the plant means what? Above the ground part of the plant. See, as it is, if all this method of introduction will fail, that time we will take the solution or the liquid of this radiolabeled compound, will spray on the aerial parts of the plant. So when it will attach to the any part of the plant, through that directly it will enter into the medicinal plant or any part of the plant. So this is the second point, introduction of this radiolabel compound into the biological system. Now what will happen, once this radiolabel compound will enter into the medicinal plant, it will directly go to the biosynthetic pathway. So from biosynthetic pathway, it will directly enter into the final compound. If it is a glycoside, it will come into the glycoside. If it is alkaloid, it will come into the alkaloid compound. Now what we have to do after that? So after that, next step is separation and determination of radiolabel compounds in the various biochemical fraction. Means what we will do here? We know, suppose we know alkaloids, Datura alkaloid, they are present in the stems or aerial part of the plant. So in this Datura plant, we have introduced our label compound. Now what is our thing next? We will take that aerial parts of the plant. We will go for separation of tropane alkaloid. Then whatever compound we have separated or isolated from this Datura, we will try to check the radioactivity of that compound. If radioactivity will be more, based on the introduced radiolabel compound, we can see the biosynthetic pathway. So this is the connection. First we will introduce, then after introduction, that radiolabel compound will enter into the biosynthetic pathway. Then we will directly go for separation of this secondary metabolites. After separation, isolated, for that isolated compound, we will go and check the radiolabel compound. And then we will try to find out the method. So tracer technique. So these things we have to do for the radio. Now how we have to study this radioactivity for this isolated compound? So these are certain techniques. With the help of these techniques, we can directly study the various types of radioactive. So like GM counter will be there, scintillation chamber will be there, gas ionization chamber, mass spectrophotometer, NMR spectrophotometer, autoradiography. So all these are the techniques, detection techniques, which we will use to study the radioactivity of the compound. So in the GM counter, just we will introduce our compound. So all these particles will be bombarded, then radioactivity will be generated, that will be measured. So based on that, GM counter will be used to determine the radioactivity of the isolated compound. So in this liquid scintillation chamber, again same, just we will introduce our compound. It will be bombarded with the radiation, but different photomultiplier tubes will be there. They will add positive and negative ions will get attracted. Based on that, the radioactivity will be counted, and we can directly conclude whether our compound is entered into the biosynthetic pathway or not. This is the liquid scintillation chamber. Gas ionization is also same. Here we will do the ionization. ionization of our isolated compound with the help of any rays, gamma rays or X-rays or beta particle, alpha particle. Then compounds again, positive and negative ions will get separated and we will get the reading about the radioactivity. So this method also we can use for determination of the radioactivity of the isolated compound. Then this mass spectrophotometer also we can use. Here mass to charge ratio mostly will be considered. What we will do in the mass spectrophotometer? Mostly the molecular mass of the compound will be measured. Means what we will do, whatever compound will be there, that we will break into the small pieces. Based on their mass to charge ratio, each and every particle of that compound will get attracted or get separated. Based on that, we will get the result. And in the mass spectrophotometer, we will get the peaks. In that peak, one peak will always have the 100% intensity. Now, what is that? That 100% intensity peak is also called as the base peak, and molecular weight of that compound will be same as that of base peak. So these things also we can identify through the mass spectrophotometer. We can identify the molecular mass of that isolated compound and mass based on the mass to charge ratio. What are the fragments of that isolated compound, that also we can identify. So it will be directly correlated with our compound. Whether whatever radioisotope we have added in that, it is coming in the final structure or not. Based on these fragments also we can identify. Again here NMR spectrophotometer. What is NMR? It is nothing but a nuclear magnetic resonance. Here nucleus of the compound will be involved. Magnetic field will be applied. Electric field itself, that nucleus of that compound itself will have the electric field. So when this magnetic and electrical field will be in comparison with the applied field, so that compound will go into the excited state. That's why it is called as the resonance. So always magnetic field will be applied here. So in this, what we have to study, NMR spectroscopy mostly will be used to identify the number of protons or number of carbon atoms into the final product. But why we have to study here? As I told, we will introduce radio label carbon or radio label hydrogen into the secondary metabolites. If it is radio label hydrogen, we will use PMR. If it is radio label carbon, we will use C13 NMR. Means based on that, we will get idea which type of carbon or which type of hydrogen is involved in the final structure. Whether whatever radioisotope compound we are introduced into the medicinal plant, it is coming in the final structure or not. That also we can study. That is the NMR spectrophotometer. And last method is autoradiography. So in this histological tissues or chromatographic plates, mostly we will prepare and we will see whether our compound is there in that or not. Or we will take the transverse section and like photographic film will be there with the help of emulsion as a detector or ionization or radiation, different radiations we will expose and we will see whether we are able to see any radioactivity on that cells or not. Mostly here intact histological tissues we will use. In the previous method, we will use the isolated compound. That is only the difference in between all these methods. Now, going towards the different tracer techniques, it is very simple to remember. First method is precursor product sequence. In this method, what we will do, whatever precursor will be there, that is the radioisotopes will be introduced into the plant material. Whatever procedure we have studied, it is nothing but a precursor product sequence. In this technique, radio label compound will be introduced into the medicinal plant. Final secondary metabolites will be isolated. It will be purified and radioactivity of that final product will be determined, and we will check whether that compound will have radioactivity or not. If it is there, means introduced compound is directly enter into the biosynthetic pathway. This is precursor product sequence. Means whatever precursor we have added, it came into the final product and that we have identified. That's it. Now, what is the, see, easily we can study the precursor product sequence or biosynthetic pathway by this method, but there one disadvantage is there. Radioactivity of the isolated compound alone is not sufficient evidence to see whether that directly it is entering into the plant or not. Sometimes, just I told, there are two types of radioisotopes. Stable isotopes will be there, radioisotopes will be. If in the nature, stable isotopes will be there in the plant. As it is, we cannot identify this. So that time, radioactivity of isolated compound is not the method to identify same technique, precursor product sequence. because stable isotopes might be present in the nature. They may be there in the medicinal plant. It may enter into the final product. So this is the case. So that is only one disadvantage. The next and most important method is double and multiple labeling. Again, in this simple thing is double and multiple means instead of one, 2 or 3 radio label compound we can introduce into the plant. Procedure will be same. We like an example. Any alkaloid will have the nitrogen atom in their structure. So that nitrogen atom will be enter into the medicinal plant. Then it will enter into the biosynthetic pathway and it will be present in the final product. Suppose in that particular structure, if nitrogen atom will be present at 2 places, and if we will add nitrogen atom as a radioisotope, it may go at one place, but we don't want that. What we want, whatever nitrogen atom present, means wherever it is present, that nitrogen atom should get replaced with the radio label compound. In that case, we will use double and triple labeling method. Means in any structure, if more than 2 or more than 1 nitrogen atoms will be present, in that case, we will add radio label nitrogen, but condition is all the nitrogen present in the precursor should get replaced with the radio label nitrogen. It is called as double or multiple labeling. Means in one compound or one precursor, 2 or 3 compounds will be introduced, but it might be same. Sometimes we may add carbon and nitrogen, sometimes carbon and hydrogen, carbon and nitrogen, any type of 2 or 3 labeling also we can do. So to just see whether same ring structure is forming by this way or not, that also we can study with the help of double and triple labeling method. Now last one is the competitive feeding. Competitive means what? See, like there will be the certain like if we'll take an example of any biosynthetic pathway, one compound will be forming by 2 biosynthetic pathways. Suppose here one example is A is the precursor, it is converting into the final product as C. But when A is converting into the C, means precursor is converting into the final product or the secondary metabolite, that time what will happen? There may be 2 types of this intermediate compound. That intermediate compound may be B or B dash. Now we have to find out whether our A to C is through ABC or through AB dash C. In that case, means see wherever competitive means there is a competition for the intermediate compound. Means A will be converting into the B will be converting into the C, or A will be converting into the B dash will be converting into the C. Means there is a competition for ABC pathway or AB dash C pathway. In that case, what will happen? Whoever is having prominent concentration, that pathway will be carried by the secondary metabolite. Now, this type of study will be done with the help of competitive feeding. So what we will do? We will add radio label compound into the A, and then we will try to see whether it is carrying ABC or AB dash C. What we are doing in the precursor, that is in the A, we are trying to add the radio label compound. So then we will see the activity of the C. If a radio label compound will not enter, we will not get activity. If a radio label compound will enter into the pathway, we will get the radioactivity. And based on that, we can see through which competition we are getting the final product, that is competitive feeding. Now, there are so many applications of the tracer technique. See, cyclization type of reactions also we can study with the help of this tracer technique. Like here we can use radio label carbon, radio label hydrogen also we can use. Best example is mevalonic acid pathway. Then we can see the any structure like a steroid is at which position, as steroidal structure is very big, C17 carbon atoms will be there. So at which position interrelation is there, that type of studies also we can do. Means how rings are forming into the biosynthetic pathway or any secondary metabolite, that study we can do. A cyclization type of reaction, how cycle is forming or how ring structure is forming into the medicinal plant, that technique or that study also we can do with the help of tracer technique. Then biosynthesis of terpenoid, that is volatile oils, how they are synthesized by the chloroplast, that is nothing but a leaves. What will happen when leaves will prepare their own food material, they'll prepare the byproducts. So that byproducts are nothing but the terpenoids, and all the terpenoids will get then deposited in various parts of the plant. But how these secondary metabolites will be synthesized, that study also will do most of the time. Then which are the intermediate compound, like formation of cinnamic acid is another example. In the coumarin pathway, see, whenever coumarin nucleus will form, as it is nothing basic nucleus for the biosynthesis of flavonoids. Coumarin is a just one basic nucleus, other than double bond O ketone group. Coumarin nucleus is same as that of flavonoid nucleus. How this nucleus is getting formed? So, but in that again, cinnamic acid will form. Cinnamic acid itself will have some biological activity. So, formation of 2 or 3 compound at a time also we can study. Means how they will study. Like I told, competitive study or competitive analysis is also there. So, these type of things also we can study with the help of biosynthetic pathways. Now, carbon and nitrogen atom purine nucleus, like we will take an example of purine based alkaloid, like purine nucleus is mostly present in the TN copy. So how these rings will form, complicated structures are forming into the medicinal plant. So that also we can study with the help of tracer technique. So there are so many, like how calcium will be like we know most of the times what will happen, we will have the calcium oxalate crystals into the medicinal plant, but how these calcium oxalate crystal will form, how they are coming, that study also we can do with the help of tracer techniques. So all these are the general introduction about the tracer technique. Here we have studied in short what is tracer technique, how we can introduce all these radio label compound into the medicinal plant, what is the general procedure, which are the techniques in short. So other than that, if you see, there are so many types of questions will be asked in any universities on this topic. So this topic is most important topic and always question will be there on this topic. Now which are the types of question? First is biosynthesis. What is biosynthesis? Synthesis of the secondary metabolites into the biological system. Biological system is nothing but a medicinal plant. So that is the biosynthesis. So definition of biosynthesis question will be always asked for the 2 marks. Then next is the define radioisotopes. Give the some example used in the tracer technique. Just I told radioisotopes are nothing but a stable isotopes or the any atom or compound when we will bombard with the radiation, we will get the stable radioisotope, and that can be used into the medicinal plant to study the tracer technique. So examples are carbon, hydrogen, oxygen, nitrogen, all that we can write. So there are various applications. Just now I told cyclization type of reaction to study the biosynthetic pathway and how this cyclization will occur into the medicinal plant. So all these type of things we can study with the help of this tracer techniques. The next important question is how will you introduce the radio label compound into the plant? Root feeding will be there, stem feeding will be there, infiltration will be there, and then direct spraying method will be there. All these are the method through which we can introduce the radio label compound into the medicinal plant. So next and most important question always will be asked on this is explain the shikimic acid pathway for biosynthesis of various secondary metabolite. So shikimic acid pathway is most important pathway and question always will be asked on this topic. Just we did not study in detail, but that is also there in the syllabus part of the technique. Then acetate malonate pathway and shikimic acid pathway, these are the 2 basic pathways used for biosynthesis of secondary metabolite. So mostly all the alkaloids, glycosides and flavonoids will be synthesized through the shikimic acid pathway, and most of the that volatile oils and all the oils, most of the time and flavonoid structure will flavonoid steroidal big structures will be synthesized. Terpenoids, big one, pentacyclic triterpenoids will be synthesized with the help of acetate malonate pathway. Then in autoradiography, just I told, instead of using the isolated compound, in the autoradiography we will be using intact cell structure, like transverse section, we will bombard it with the radiation, we will check the radioactivity of that cell particularly. So these things we will do in the autoradiography, and this is one of the technique where we will this technique mostly we will use to study the radioactivity of the compound into the medicinal plant. So these are the important questions mostly, and other than that, like write a note on the tracer techniques. So this is also the favorite question of all the examiner. Most of the time question will be asked, write a detailed note on the tracer technique and how we have to use that in the generation or to study the biosynthetic pathway. Then explain in detail the biosynthesis of any one secondary metabolite through the acetate malonate pathway. So for here you can write the examples of how the steroidal compounds will be synthesized, how pentacyclic triterpenoid compound will be biosynthesized into the medicinal plant. So these are the important things that you can write, and this question will be always for 10 marks. A tracer technique question will be always for 8 to 5 marks. The next and most important question is same. Explain in detail biosynthesis of any secondary metabolite through the shikimic acid pathway. So here also we can use or we can write the biosynthesis of all the alkaloids or glycoside or flavonoid compound, how it will be synthesized through the shikimic acid pathway. So that in detail biosynthetic pathway should be written. So here basic biosynthetic pathway we have to write, that is shikimic acid pathway we have to write, and that again it will be carried to the final biosynthesis of secondary metabolite any compound. So 2 pathways here we have to write, and this question will be always for 10 marks. Then write about the precursor product and sequential analysis method in the tracer technique. Just I told precursor product sequence is very simple concept. Just we have to use the precursor as a radio label compound. It will be introduced into the precursor that will enter into the biosynthetic pathway, and through the biosynthetic pathway that radio label compound will be introduced into the final product. So that final secondary metabolite will be isolated and radioactivity will be measured. That is precursor product sequence. Next is the sequential analysis, like A compound will be converted into the B will be converted into the C. So that sequence of the compound is same or not, that also we can study. So just we will do what, just one, we will add a radio label compound in radio label compound in the precursor, that is A will introduce into the plant, it will convert into the B, A and B dash will be there, and then it will come into the C, that is A, B and C dash, like that. So this is the sequential analysis, sequence of the biosynthesis of that particular compound we can study in this method. So next and most important question is write about the role of radioisotopes in the investigation of biogenetic pathways. So what is the role? All these radio label compound can be introduced into the medicinal plant so that it will enter into the biosynthetic pathway, and radioactivity of that compound will be studied with the various method. So the actual role is just it will give the idea about how that biosynthetic pathway will be there in the medicinal plant. Just we are tracing the biosynthetic pathway of the secondary metabolite by using the radioisotope. So radioisotopes will be formed by the 2 methods. One is as it is, it will be available in the nature from the stable source, and other one is with the, we will bombard that with the radiation. Then last question in this will be the write in detail about the amino acid pathway. So amino acid pathway, it is a general basic biosynthetic pathway that we have to study here, and this biosynthetic pathway will be there always. Like how amino acids will be synthesized. There are various types of amino acid. Actually, first amino acid pathway we have to study. In this, so many amino acid first will be synthesized into the medicinal plant, and all these like tryptophan, tyrosine, phenylalanine, when they will synthesize into the plant body, all these amino acids will be carried for the shikimic acid pathway, precursor product sequence, and then when they will enter there, from that only we are getting the secondary metabolite. So basic amino acid pathway is the first pathway that we have to study. Then that whatever amino acids will be synthesized into the medicinal plant body, that will be enter into the shikimic acid pathway and acetate malonate pathway. So that amino acids will be converted into the shikimic acid will be converted into the various secondary metabolites. So all these are the first 3 basic pathways we have to study, that is amino acid pathway, shikimic acid pathway, acetate malonate pathway. And then from that directly we have to go for the synthesis of this secondary metabolite. So all these things we have to write in detail. So in this topic today we have discussed the methods also. There are various techniques for this tracer, how to study the biosynthetic pathway, tracer technique, sequential analysis and quantitative feeding. So these are the 3 important methods which we have to study for the biosynthetic pathway, which are the application that we have to remember. And then how we have to frame this radio label compound, which are the methods. Just we have to bombard all these compound with the radiation, alpha, beta or gamma radiation, and based on that only we will get the stable radioactive compound that we have to introduce into the plant body. So this is is the important things. Now, introduction of all these secondary metabolites on introduction of this radio-label compound into the secondary metabolites also we have to study. So all these are the general introduction about this topic. And we have studied the biosynthesis also. Biosynthesis means synthesis of the secondary metabolite into the medicinal plant. And here we are using the biological system that is nothing but a medicinal plant. So this is just an overview of, in short overview of this topic, tracer technique. So we have to study all this topic in detail. We have to see the amino acid pathway also in detail, shikimic acid pathway, acetate malonate pathway also we have to study in detail. And a big pathways will be very big. First we have to study the basic pathway, and from that how we are getting the synthesis, or how this shikimic acid or acetate malonate, they are converting into the big structure that is secondary metabolites. That also we have to study in this topic. So all these are the questions on this topic, and most of the questions, or other than this, no other question will be asked on this topic. So this is just an short overview of this one, tracer technique.